Welcome to this short presentation being given by Boyds. I'm Alan Boyd, and I'm going to be joined in this session by Dr. Nick Myers, who's the Vice President of Product Development at the company. At Boyds, we help companies and universities translate their ideas into medicines. And during this session, we're going to talk briefly about how we go about this process. No matter what the product is, and you can see on this slide now that within pharmaceutical medicine, we will develop medicinal products, these tablets, biologicals, advanced therapies, but also medical devices and diagnostics. But no matter what the final product is, we have to undergo a very similar process in turning that research ideas and those discoveries into medicines or the devices or diagnostics or a combination of all three. Now, development is a multidisciplinary approach. It's not just about getting the clinical trials done, but it needs to cover how the product is going to be manufactured and the regulations around that, the manufacture, any preclinical work that needs to be undertaken, including proof of concept work that gets done either in vitro or in vivo, and also any animal toxicology that's needed for the product. And then finally, there's the clinical trials, and to demonstrate and prove and confirm that the product is actually safe and effective in the indication that you're looking for as a treatment to help treat patients. And over it all, we have the regulatory activities because we cannot develop a medicine without the input from the regulatory authorities such as the MHRA in London, the FDA in the States, and the EMA. On this slide, we've given a schematic of the development process. And you can see that it's built as a pyramid, and that's because we base it upon doing the preclinical work to start with, as I've described, like the toxicology and the in vivo and in vitro proof of concept work, and then gradually move into clinical studies, as you've seen here with phase 1, 2, and 3 clinical studies. And this is all built up together to allow registration of the product. And during this process, we'll cost it out and work on a timeline to make everything is happening in sequence to make sure that we do achieve that registration process at the top. Now, to start off with all this, clearly we need to know what sort of product we're developing. This is where what we call the target product profile comes in. And the target product profile is a summary of all the activities and what the product needs to look like for use in patients later on. And this is no different than, say, if you're developing and building and designing a car or a washing machine, you need to know what the final product would look like. And ultimately, the target product profile through the development process will subsequently turn into the prescribing information, the summary of product characteristics for use by healthcare professionals, and also the prescribing information, patient information leaflets for patients. The other advantage we've got as well is that when we're developing treatments like advanced therapies, then we don't need to do all the activities because we typically go straight to patients. So therefore, the development process is a bit shorter and is an example really of how we use the total development process to see what we need to do and perhaps more importantly, what we don't need to do. On this slide, I've outlined the content of a target product profile. You can see we cover things such as the anticipated registered indication, the therapeutic area, thinking about the route of administration and the dosage form, and also think about the patients themselves that we're actually going to be treated in terms of the patient profile, how many patients there are around the world, and what current therapies are there. And building onto that, then we think about the regulatory strategy and our customer groups and how we're going to talk about this product to healthcare professionals. And at the same time, we'd also draft a typical prescribing information around what the product may eventually look like. So we put these things together to help steer and guide the development process.
In the rest of today's presentation, we're going to talk about the early development process through to a first-in-human clinical study, because unfortunately we don't have time to cover the entire development program through to approval and launch. So during the candidate selection and candidate profiling stage, there are various activities that need to be done. Critical is demonstrating that the product will work. In vitro and in vivo proof of concept studies will show whether the expected mechanism of action is happening and give confidence that one can ethically take a product through to a clinical study in humans, which is actually going to work. This is also a period of time in which invaluable information can be generated in animal models to look at the pharmacokinetics, to look at the acute toxicological effect on cells, if there is any, the potential for immunogenicity, and also to begin to develop the assays that are going to be required later to measure the drug in cells and tissues and in the bloodstream, and also to look at the stability of the drug systemically and potentially locally, depending on the route of administration. Once the decision has been taken to take a candidate product into full development, one has to consider the CMC or quality, the non-clinical and the regulatory aspects which need to be paid attention to in order to be able to file a clinical trial application or to open an IND in the United States. On the CMC side, one will start to look at the manufacturing process and to optimize that process, particularly thinking about aspects such as, is the manufacturing process likely to be scalable as we work through the later phases of clinical development? Will the starting materials be readily available, and what are the cost of goods likely to be? One needs to develop and validate to the required extent, depending on the phase of development, the analytical methods that will be required to release the product. Preliminary stability information will need to be generated to support clinical trials. Now, depending on how many applications of the drug there will be in early development, that may only require 1 month stability or maybe 3 months stability. But that data has to be generated from ICH compliant stability studies. And one will also be able to undertake preliminary formulation development. The formulation that's often used in phase 1 studies, particularly in healthy subjects, may be very different from the formulation that ultimately will be given to patients in phase 3 trials. The non-clinical program will vary greatly depending on the type of product one is developing and also on the clinical indication. These factors will impact the selection of the species for toxicology studies and also whether GLP or non-GLP toxicology studies are required, which also have an impact on where it's possible to do those studies. For example, with many gene and cell therapy products, it's perfectly feasible to do non-GLP studies within a university or academic type environment, as long as the appropriate checks and balances are put in place. And there may also, depending on the product type, be the need for some preliminary genotoxicology work prior to a first-in-human study. And then, of course, the regulatory piece. Whatever the non-clinical program is that one proposes to do, this needs to be sanity checked with the regulatory agencies, either at scientific advice in Europe or a pre-IND meeting with the FDA in the US. It may be also an opportune time to look at special designations. If we're talking about a rare disease, can an orphan drug designation application be made? Is there a case to be made for applying for prime designation in Europe or breakthrough designation in the United States, which can help to accelerate development and give more frequent and in-depth access to the regulatory agencies? And then, of course, the preparation of the clinical trial application or the IND itself, which is not a light undertaking. Probably one needs to dedicate 6 months to preparing these regulatory filings. And then finally today, just to give you some tips about development planning. Planning is critical. It's going to be important to develop a realistic plan, which includes the resource allocations, timescales, and costs. It sounds obvious, but work forwards, not backwards from a corporate goal. It's very difficult to shoehorn a development plan in to a pre-existing window of time. The experts have to be consulted on how long various studies and activities will take, and then you can build a realistic plan. Also, building contingency plans. Think about what could go wrong and what you will do if you have to change direction. I cannot underestimate the importance of early dialogue with the regulatory agencies, and also to sanity check what's happening in the program. Convene independent scientific advisory boards to interrogate the data which which is being generated. This is important to avoid developing your product within a bubble or developing groupthink where everybody thinks the product's fantastic and you can't actually see the issues within the data. And revisit your TPP frequently. The TPP is a living document and may have to be modified depending on the competitive environment. And also in the lead up to the clinic, engage your key opinion leaders in the field and look at the clinical and the unmet medical need to see if those things are changing. It's important, particularly in Europe, where the product needs to be priced appropriately to achieve reimbursement, that the product delivers benefit, which is more than just a tiny incremental improvement on the standard of care. So these are just some tips about development planning, things to think about during the early development phase. So I thank you for your attention. It's been a pleasure speaking to you, and Alan and I will now be available for any questions. Thank you.